Assalamualaikum and hi to Dr. Prasita and all friends. We are from Group 8. We'll be presenting hypoxia condition in lung cancer. Our group members are Nur Fadilah, Nurul Aisyah, Nur Sakinah, Nur Shazwina and Nur Ain. Okay, first and foremost, what is lung cancer? Lung cancer can happen due to the abnormal cells that clump or cluster together to form a tumor. Unlike normal cells, these abnormal cells, or we call it cancer cells, will grow uncontrollably and will disturb and destroy healthy lung tissue around them. The tumor that forms from these cancer cells is known as malignant tumor that will prevent some of our organs from functioning properly. In Malaysia, lung cancer is the leading cause of cancer-related death. According to the World Health Organization that has been reported in 2014, following breast cancer, lung cancer is the country's second most common cause of death from cancer. Next, we proceed to the causes of lung cancer. Generally, lung cancer happens due to the smoking and tobacco products. Apart from that, the other factors that can lead to lung carcinogenesis are radon gas, asbestos, exposure to air pollution, and from chronic infection. There are two classes of lung cancer. The first class is small cell lung carcinomas, which include small cell carcinomas and combined small cell carcinoma. The second class is non-small cell lung carcinomas, which include adenocarcinoma, large cell carcinoma, and squamous cell carcinoma. There are several options that can treat lung cancer, which are surgery, radiation, chemotherapy, and targeted therapy. Next, I will pass this presentation to Nur Shazwina.
Okay, hi, I am Nur Shazwina and I will explain on the terms mentioned on this topic. Okay, first one is hypoxia. It is a significant microenvironmental pressure seen in the vast majority of solid tumors. Due to rapid growth of tumors, the availability of oxygen supplies decreasing to hypoxic level below 2%, which also indicates that high proliferation of tumor cells causing the oxygen depletion. In non-small cell lung cancer and hepatocellular carcinoma, hypoxia stimulates the WNT beta-catenin pathway. Hypoxia also are mediated by the hypoxia-inducible factors HIF1 alpha and HIF2 alpha, where it will activate the transcription of genes that implicated in tumor angiogenesis. Okay, next is placental growth factor PLGF. It is a member of the VEGF family, a ligand for VEGF-R1. PLGF displaces VEGF-A from FLT1, which releases VEGF-A and allows it to activate FLK1 and enhance VEGF-driven angiogenesis. In lung adenocarcinoma cell, PLGF is highly expressed in hypoxic condition.
Hi, my name is Sakinah and I will talk about the overview of what causes the lung adenocarcinoma to progress. The cause of tumor progression in lung adenocarcinoma is the microenvironment, which is in this case is the hypoxic condition, a condition where there is a low concentration of oxygen. In hypoxic condition, there is an upregulation of HIF1 alpha that modulates the WNT beta-catenin signaling by enhancing the beta-catenin activation. HIF1 alpha also overexpresses placental growth factor PLGF that plays a crucial role in angiogenesis. Both activation of WNT signaling and angiogenesis leads to the tumor cell proliferation and growth. As the tumor keeps on growing, the surrounding oxygen is depleted, making the microenvironment to become more hypoxic.
Next, we move on the pathway that caused the cancer to grow. So first, the abnormality that caused the cancer to grow in terms of microenvironment is due to the hypoxic condition. This hypoxic condition, which later will induce upregulation of HIF1 alpha, and then it will further enhance the beta-catenin activation. This activation will modulate the Wnt signaling pathway. Upon ligand stimulation, this beta-catenin will accumulate and then will be translocated into the nucleus. Therefore, it will activate the downstream of the gene. Okay, so here are the Wnt signaling pathway that play a role in promote the cell proliferation and survival. It involves a cyanonickel pathway, which is a beta-catenin dependent pathway. The presence of xenonickel Wnt ligand will cause the beta-catenin phosphorylation and degradation is inhibited. Gene expression downstream is regulated by controlling the amount of transcription co-activator beta-catenin. During Wnt-on state, Wnt ligand will bind to the FZD receptor and LRPC co-receptor, and it causes a cascade event is initiated. The ligand binding will resulting in the disassembly of the destruction complex that stabilizing the beta-catenin. And then the signaling cascade is initiated when DSH is activated through the DIX and PDZ domain and FZD associated distributor protein. After activation of DSH, it will bind to axin, which later will inhibit the GSK3 phosphorylation of beta-catenin in the destruction complex. Therefore, in the absence of destruction complex, beta-catenin will accumulate in the cytoplasm, eventually in the nucleus, and finally will activate the transcription factor, which later will promote cell proliferation and survival.
Next, the treatment for lung adenocarcinoma. The first one is silencing PLGF. The expression of PLGF can be knocked down by using lentivirus-mediated RNA interference. Listed are 3 lentivirus-mediated RNA interference being used in the journal. The word SH in front of each PIGF represents the short hairpin RNA, which is an artificial RNA molecule with a tight hairpin turn that can be used to silence target gene expressions via RNA interference. They target mRNA cleavage, therefore can repress PLGF expressions at both mRNA and protein levels effectively. Not only that, through this method, expression of c-MYC can be downregulated, thus reduce the stimulation of cell proliferation and glycolysis of lung cancer cells by hypoxia. c-MYC is encoded by an oncogene MYC. c-MYC is a transcription factor involved in cell proliferation, apoptosis, and metabolism that contributes to tumorigenesis. Inhibition of Wnt and beta-catenin pathway is another treatment to treat lung adenocarcinoma. A potent inhibitor, which is XAV939, acts as a therapeutic agent that selectively inhibits 10-kinase family members. 10-kinase is expressed in lung cancer cells in high levels and promotes glycolysis via activation of Wnt and beta-catenin signaling. Shown is a simple pathway of the inhibition. First, it blocks the expression of 10-kinase and it stabilizes the axin expression, thus it will disrupt the beta-catenin formation. Therefore, through this method, 3 factors can be reversed, reducing the cell proliferation and glycolysis of lung cancer, which are PLGF overexpression induced cell proliferation, expression of beta-catenin, and protein expression of c-MYC, and also lactate dehydrogenase A.
As a conclusion, this research indicates that by inhibiting the WNT beta-catenin pathway, it will reduce the activation of cell proliferation and glycolysis that has been triggered by hypoxia. Apart from that, PLGF can be identified as the possible therapeutic target in lung cancer treatment. These are the list of reference that has been used in our assignment. That's all from our group. Thank you.